Chapter 11, Section 2, the process of cell division. Learning objectives: explain the role of chromosomes during cell division, describe the main events of the cell cycle, describe what happens during the phases of mitosis, and investigate how daughter cells split apart after mitosis. Cell division takes place all of the time, keeping us healthy by replacing worn cells and regenerating the tissue we lose during injury or disease. What would happen if a cell were simply to split into two without any advanced preparation? The result might be disastrous, especially if some of the cell's essential genetic information wound up in one of the daughter cells but not in the other. In order to make sure this doesn't happen, cells first make a complete copy of their genetic information before cell division can begin. Chromosomes are genetic information that are bundled into packages of DNA. In eukaryote cells, DNA is packaged into multiple chromosomes. DNA bound around proteins that are called histones are called chromatid. Chromosomes are precisely separated into two daughter cells during cell division. A prokaryotic cell lack a membrane-bound nuclei and many of the organelles found in eukaryote cells. The DNA molecules of prokaryote cells are found in the cytoplasm, along with most of the other contents of the cell. Most prokaryotes contain a single circular DNA chromosome. That circular DNA contains all or nearly all of the genetic information. In eukaryote cells, they generally have much more DNA than prokaryotes, and therefore they contain multiple chromosomes. Fruit flies, for example, have 8 chromosomes. Carrots have 18. Human cells have 46. The chromosomes in eukaryotic cells contain DNA tightly wound around histones. The complexity of the DNA and protein is referred to as a chromatid. In the prokaryote cell cycle, the prokaryote cell is a regular pattern of growth, DNA replication, and a cell division that can take place very rapidly under ideal conditions. The process of cell division in prokaryotes is a form of asexual reproduction known as binary fission. Once the chromosomes have been replicated, the two DNA molecules attach to different regions of the cell membrane. A network of fibers forms between them, stretching from one side to the other side. These fibers constrict and the cell is pinched inward, dividing the cytoplasm and chromosomes between the two new formed cells. Binary fission results in a production of two genetically identical cells. Eukaryote cells have a much more complex cell cycle than prokaryote cells. During the cell cycle, a cell grows, prepares for division, and then divides to form two new daughter cells. Interphase is the time in between cell divisions and is divided into G1, S, and G2 phases. During G1 is when cell growth occurs. Cells do most of their growing during the G1 phase. In this phase, cells increase in size and synthesize new proteins and organelles. The G in G1 and G in G2 stands for gap, but the G1 and G2 phases are actually periods of intense growth and activity. The S phase is DNA replication. The G1 phase is followed by the S phase. The S stands for synthesis. During the S phase, new DNA is synthesized and the chromosomes are replicated. By the end of the S phase, the cell contains twice as much DNA as it did in the beginning of the phase. G2 phase is preparing for cell division. When DNA replication is completed, the cell enters the G2 phase. G2 is usually the shortest of the three phases. During the G2 phase, many of the organelles and molecules required for cell division are produced. When the events of the G2 phase are completed, the cell is ready to enter the M phase and begin the process of cell division. The M phase of cell cycle, which follows interphase, produces two daughter cells. The M phase takes its name from the process of mitosis. During the normal cell cycle, interphase can be quite long. In contrast, the process of cell division usually takes place pretty quickly. In eukaryotes, cell division occurs in two main stages. The first stage is the process of division of the nucleus, and it's called mitosis. The second stage, the division of the cytoplasm, is called cytokinesis. In many cells, the two stages may overlap so that cytokinesis begins while mitosis is still taking place. Biologists divide the events of mitosis into four phases: prophase, metaphase, anaphase, and telophase. Depending on the type of cell, mitosis may last anywhere from a few minutes to several days. The first phase of mitosis is prophase. During prophase, genetic material inside the nucleus condenses and the duplicated chromosomes become visible. Each duplicated chromosome condenses to appear as two thick strands known as sister chromatids. They are attached at a point called a centromere. When the process of mitosis is complete, the sister chromatids will have separated, one to each of the daughter cells. Also, during prophase, the cell cycle starts to build a spindle, a fan-like system of microtubules that will help to separate the duplicated chromosomes. Spindle fibers extend from a region called the centrosome, where tiny paired structures called centrioles are located. Early in prophase, the centrioles move towards opposite ends or poles of the cell. Plant cells lack centrioles and organize spindles directly from their centrosome regions. The second phase of mitosis is metaphase, and it's generally the shortest phase. During metaphase, the centromeres of the duplicated chromosomes line up across the center of the cell. Spindle fibers connect the centromere of each chromosome to the two poles of the spindle. The cell is now ready to separate those sister chromatids. The third phase of mitosis is anaphase. This begins when sister chromatids suddenly separate and begin to move apart. Once anaphase begins, each sister chromatid is now condensed as an individual chromosome. During anaphase, the chromosomes separate and move along spindle fibers to opposite ends of the cell. Anaphase comes to an end when this movement stops and the chromosomes are completely separated into two different groups. Following anaphase is telophase, the final phase of mitosis. During telophase, the chromosomes, which were distinct and condensed, begin to separate and spread out into a tangle of chromatid. A nuclear envelope reforms around each of the cluster of the chromosomes, and gradually a nucleus becomes visible in each daughter nucleus. Mitosis is complete, but the process of cell division has one more step. As a result of cytokinesis and mitosis, two nuclei, each with a duplicate cell of chromosomes, have formed. All that remains to complete the M phase of the cell is cytokinesis, the division of the cytoplasm to form two separate cells. Cytokinesis usually occurs at the same time as telophase. Cytokinesis completes the process of cell division by dividing one cell into two. The process of cytokinesis differs in animal and plant cells. In animal cells, during cytokinesis, in most of the animal cells at least, the cell membrane is drawn inward until the cytoplasm is pinched into two nearly equal parts. Each part contains its own nucleus and cytoplasmic organelles. Cytokinesis in plant cells proceeds differently. The cell membrane is not flexible enough to draw inward because of the rigid cell wall that surrounds it. Instead, a structure known as a cell plate forms halfway between the divided nuclei. The cell plate gradually develops into cell membranes that separate the two daughter cells. A cell wall then forms in between the two new membranes, completing the process. There's a picture of this on page 504 of your textbook. Mitosis overview. You should be able to list and describe the phases of mitosis. Interphase, which is prior to mitosis, is when the cell grows and replicates its DNA and centrioles. The first phase of the M phase, or mitosis, is prophase. The chromatin condenses into chromosomes. The centrioles separate, and the spindle begins to form. The nuclear envelope breaks down. The second phase is metaphase. The chromosomes line up across the center of the cell. Each chromosome is connected to spindle fibers at the centromere. The third phase is anaphase. The sister chromatids separate into individual chromosomes and are now moved apart. The fourth phase and final phase of mitosis is telophase. The chromosomes gather at opposite ends of the cell and lose their distinct shapes. Two new nuclear envelopes will form. And then finally cytokinesis. The cytoplasm pinches in half. Each daughter cell has an identical set of duplicate chromosomes.